Get this party started. Hello, everybody. I'm Anna Tanasa. I'm an integrative pediatrician in Tucson. I know most of you. Thank you for coming tonight and sharing space with us. Tonight, we have the privilege of having Dr. Russell Witte. He's a professor of radiology, optical sciences, and biomedical engineering, neurosurgery at the University of Arizona. Dr. Russell Witte received his bachelor degree in physics from the University of Arizona and his Ph.D. in bioengineering from Arizona State University in 2012, where he made key discovery related to neural coding and learning-induced plasticity. After a 5-year postdoc experience in the Biomedical Ultrasound Imaging lab at the University of Michigan, he returned to his hometown, Tucson, in 2007, joining faculty in the Department of Radiology, Optical Sciences, and Biomedical Engineering. As a professor at the U of A, Russell continues to develop cutting-edge hybrid imaging modalities that integrate light, sound, microwave, electricity to address grand challenges in medicine with applications that extend from head to toe. Russell has directed large research projects funded from NIH, NSF, DoD, and DARPA, published more than 200 reference publications, holds over 12 patents, and is founder of ElectroSonix, a startup dedicated to revolutionary technology to map the heart and brain at unprecedented resolution. So for tonight, we're going to have a presentation of 45 minutes. Then we're going to stop the recording and do question and answers. We'll have a sign-up sheet for everybody that's present if they want to stay in touch with us. We'll try to record it and put it on our Facebook page for future references or for people that couldn't make it tonight. So here we are. We have Dr. Russ.
All right. Thank you so much for the introduction. And I'll tell you, it's a little weird feeling being a few feet from where you were born. So it's somewhere around here. I probably should recognize the room. So I need to ask my mom, who's not here today, but I just saw her. So Anna asked me to specifically talk about nanotechnology. And although I've worked with nanoparticles and nanotechnology, that's not necessarily central to what I do. So I have one slide about the kind of things I do at the university in my research. And it's really about coming up with new ideas, new imaging modalities that use these types of energies that Anna mentioned. And almost everything is related or uses sound or ultrasound in some way. So I'm kind of giving you examples of the kind of things you can image using ultrasound. And some of these are combined with other forms of energy like microwaves to look at temperature, dielectric properties. We look at electrical currents with ultrasound, a kind of a unique modality here called acousto-electric imaging that we're developing in my lab for brain and heart imaging. Photoacoustic imaging, which uses light to generate sound and make ultrasound images that's related to the absorption of light. And then another example here that I have in the slide is looking at the elastic properties using ultrasound to advanced signal processing. So that's really the only slide that's central to the kind of things that we do in my lab. And you can tell that not necessarily there's any nanoparticles in there, although the photoacoustic imaging one does have a dye. And I have worked with nanoparticles and dyes before. I prefer not to use them because if we can be completely non-invasive and use intrinsic properties of the tissue without using contrast agents, it's even better, especially when we talk about translation. And hopefully by the end of this talk, you'll kind of see why we may want to not embrace kind of nanoparticle technology based on what we know now. Although we can, so what I want to just start out with is always thinking about the purpose and use of any technology and kind of where that takes us kind of in the future. So if you look back in the 1990s when I was in graduate school, these were kind of the hot headlines referring to the future of medicine, referring to genetic engineering, and this is 15 years before CRISPR technology, and also the idea of cloning. Okay, so that was kind of what was in the headlines. However, when you look today at healthcare in the United States, it is not a very good picture when you simply compare us to kind of peers, especially Europe and other first world countries. By far, we pay much more per person in terms of healthcare per year, and our life expectancy is pretty much near the bottom. So this should be the impetus of any kind of conversation about any new technology or ideas that to address bringing people together, because there's each of these axes is addressing two different things on that plot. One related to the economy and the dependence on having sick people. And then as this slide points out, 18% of our GDP is devoted, or maybe my next slide, or maybe I had it on the previous slide. Yeah, 18% of our GDP is dedicated to taking care of sick people one way or the other. However, the results are not that favorable when you kind of look at almost any category you want. You can look at chronic diseases, you can look at infant mortality, you can look at maternal mortality, you can look at chronic diseases in children. I'm just putting a few stats up here. Usually I have many more slides that we can get into. And this promise of having Alzheimer's disease double every 15 years is not a necessarily optimistic outlook, but that's typically what we're told in terms of research, and we've got to prepare for that. Not like we can do anything, we just have to manage and prepare for it. That's usually not how I think, because I like to solve problems. So even taking that sort of attitude doesn't make a lot of sense to me. And this is the impact in terms of the economy. I mentioned the 18% GDP. This is just how much we spend on prescription drugs in the United States. And yet prescription drugs, taking them normal as prescribed, is the third leading cause of death. And it's really much higher than the number there in that bottom bullet. And so just some simple questions that we want to think about and look at is, how much does industry influence medical science? And I really like to look at history, and I'm not going back too far with my slides in this talk. I generally like to go back to 1900, but we can look at the examples from smoking and these in particular, where it was actually promoted for pregnant women to smoke, that it was good for their health. And that these were considered facts, and the CDC didn't see any problem, even though we know since the 1800s there were all kinds of problems with tobacco and smoking that was out there, publicly available information. And you even had children advertising smoking, and that's an ad from Stanford University in their archives. Another thing to consider before we get into the nano side of this is that Goldman Sachs was asked in 2018 whether curing disease is a sustainable business model. And they cited the example of Gilead Sciences coming up with almost a cure for hepatitis C. And in doing so, they reduced the incidence 90%, and they lost $8.5 billion in sales in so doing. And so their conclusions was that that's not a really good way. Looking for chronic therapies is a much better way for companies to pursue long-term profit. Okay, so something to keep in mind, the influence of industry. Now, this is the typical kind of model from, and you might see in a biology book, image of a cell and the periodic table, because we normally think of that as the starting point for chemistry. So I'm using this as the traditional conventional model that we use today. And because nanoparticles are thought of as built up from atoms, and the chemists are the ones that normally would be designing and developing these, this is kind of the model that we want to work with. But before I get into the nano, we want to look at a couple other things, because there's really a part two to this talk that I'm not going to have time to go over. And that's going to be related to thinking in terms of waves, frequency, scale, resonance, vibration. I can put in a lot of other numbers. And the examples I showed from my imaging using ultrasound and light, you know that I tend to use all these types of energies, and it's important to think that way. But the goal of this talk is to focus in that nano box there, which is a tiny window. And if you think of wavelengths of a wave, then you can relate the nanostructures to that little box there. And you can see we're between the scale of a cell and an atom, somewhere in there. But we're kind of taking a sliver and making a big deal out of it. Now, it's a big deal because it's right below of kind of what we can typically see under a microscope. So people got very excited, specifically Richard Feynman in 1959, who gave the famous presentation at Caltech. But that's really what we're going to be focusing on, is that little window. But it's really a window to a much bigger system that we can think of as both spatial and frequency domains here. Now, that biochemical model isn't the only model that's been around. In fact, that's technically a modern, more modern model of looking at the human body. And after World War II, it's important to note that this biochemical model was prevalent in universities and academia, as well as military and classified research. And we know this as biochemistry, cellular and molecular biology. And that was the direction that especially universities went for the most part. However, there's a whole other field before World War II and then after World War II when you get into military classified research. And that's really looking at the body as a bioelectrical system, a biophysical system, and the role of electricity and magnetism and its impact on biology and the human body. And this article from Columbia University in 1939 talks about how we understood that the body operated like a radio station. And so you can kind of read that. And this is an important divide because if you actually look back, and this is going to be really part 2 of a talk if I come back, because this is typically what I talk about in terms of thinking about therapy and treatment in terms of energy medicine or vibrational medicine. Back around 1900, this was prevalent. And you look at the Sears Roebuck catalogs back then, you had pages and pages of electrical devices that people could buy and use at home, from the violet ray, like I'm showing there on the left, that people still use today, that was patented by Nikola Tesla, to many other types of devices. And back then, it was seen that the medicine of the future, and you can see in that little clip from the 1800s, was the medicine of the future, electricity. And these are kind of Bibles of how currents were used for treatment from every type of your body, from head to toe, from your teeth. These are thousands of page documents and often written by MDs and PhDs. So I'm kind of giving you examples of things you can find. And when I say high frequency currents, you might not be obvious what those are, but typically those are currents that are below 100 kilohertz. And engineers and physicians at that time were meeting routinely at conferences to share ideas of technology and how they were impacting their patients. So this was not just a fringe thing, not just in the naturopathic community. In fact, more than half of allopathic doctors around 1910 were using electrical devices for treatment. Now, there's a whole history of that I won't be going into in this talk, because you'll have to invite me back for more to figure out kind of what happened and where we are today. But I'm just going to give you a couple of glimpses here, including Albert Abrams, who is Dean of the College of Medicine at Stanford, well known for his pathophysiology books, and also considered the father of vibrational medicine, the founder of radionics, which could be considered a controversial topic. But he was a pioneer at the time. Today, some people refer to him, despite the titles up there, as the Dean of Quackery. So depending on how you look at things, you already have a controversy here developing. I will not go into detail because I just want to get to the nanotechnology side here. So you also have George Lukovsky and his healing coils, electrical coils that use potentials there. You can see the little sparks coming off of these. These devices were used in hospitals across the United States and Europe for decades. And were slowly phased out after he died around during World War II. But he and many others were able to show that these types of principles, in these cases, you're looking at treatment of originally testing plants that could develop human-like tumors that could be healed by using these coils, and then also taking this to patients. Royal Rife was an amazing optical scientist and bacteriologist, 1920s to 1940s. He was recognized as the microscope of the decade, along with the electron microscope in 1940. He built a super microscope that was able to overcome the diffraction limit and see living organisms in culture as small as 50 nanometers. And that's one of the amazing feats that he did within his efforts to look for a virus that was connected to cancer, because at that time people thought infectious diseases all had a origin of some sort of parasite, virus, or bacteria. And since they couldn't find the cancer virus, he set out a mission to build a microscope that could get down to that scale. And there's a whole history to this. And this is the microscope side. There's another whole history to Royal Rife when he came up with resonant therapies principles to actually eventually do clinical trials at University of Southern California, using principles of how the microscope worked and then applying that to resonant frequencies using cathode ray tubes So there's documentaries you can find in more detail about that. And then finally, in a more modern time, when we talk about the electrical system, and at this point, there's no nanoparticles. This is using waves. This is using frequencies. When we introduce nanoparticles that can accentuate the effects of electricity and magnetism, but just by themselves, these energies can have therapeutic effects. And these are two of the pioneers in that field, certainly when we talk about the 1960s and onwards. Bjorn Nordstrom was on the chairman of the Nobel Assembly and did clinical trials in treating cancer and tumors. He's most well known for the inventor of the biopsy needle. And he was ridiculed when he came up with that idea and said no one was going to be sticking needles in people. And of course, today that's routine. But the other side of things, he made amazing discoveries with how your body is networked electrically. And then Robert Becker, who's probably one of my favorite orthopedic surgeons, discovered ways of using electricity to completely regrow limbs. He was the first one to do that in a frog back in the 60s, published that in Nature. And he was a mainstream person in the sense that he was at Syracuse University with NIH Navy funding and published in the top journals. But still, half of the orthopedic surgeons I talk to are not familiar with his research, which is somewhat shocking. So finally, the motivation for what I just showed you is to think twice about things that were going on even before 1953. This is the Fitzgerald report to Congress when he was asked to investigate rampant reports of medical suppression in the United States and beyond, but primarily the United States, looking at the AMA and the FDA in particular, and the journals that were associated with them. And you can clearly read the quote there in the middle. But basically, the findings were that the suppression of medical research in this country was rampant and out of control at that time, and he provided the documentation related to that. So then you have to question what were the discoveries that were being made that were being suppressed, and then did Congress then say, okay, let's get a handle on this and end the medical suppression? Well, the answer is absolutely not. The Politics and Healing, written by Daniel Haley, who is a state senator at New York, talks about the backroom conversations talking about medical suppression and the kind of things that are going on. So there's a long history and story to this, but it's certainly relevant when we start talking about looking at the past and history to come up with remedies for the present and future. All right, so now we get into nanoscale talk. Okay, so I mentioned Richard Feynman gave this lecture in 1959 at Caltech. He kind of gave a second lecture in 1984, which you can actually see online. But a lot of the things that he talks about in that lecture and his vision are things that I'm going to be talking about in modern times. And it wasn't just biology and medicine, but some of it was biology and medicine. So first, let's get an idea of scale. So it's really important to understand what nanoscale is. And it's 1 order, 2 orders of magnitude bigger than an atom, okay, a minimum. We're talking, so we're in that window of 10 to 100 nanometers. Yes, we can go up to a micron, but NIH often thinks of things on the 100 nanometers as a scale smaller being nano. But regardless, that's kind of, you got 2 orders of magnitude you can work with between an atom and a cell, I suppose, 1 to 2, maybe 2 to 3 orders of magnitude. Okay, so keep that in mind. And I just throw this in there to think about that the physics is so different. Usually I'll show like cell motility. I took that out, but the physics when you get down to the nanoscale is very different than what you might feel when you're walking around where gravity kind of dominates. You know, and you hit something, the forces that act on us in the macro world are completely different when you get to the nano world. Partly because, especially when you have buoyant forces, so you got tiny particles that are suspended in solution, it makes them basically almost weightless. And the things that will dominate are proximity and electromagnetic forces. So you have to kind of think that the world is very different when you go to the nanoscale. And here, these are things, many of these I've worked with before, but it gives you an idea of the scale when we talk about nanoscale, and I'll talk about the word smart in a second, but these platforms, you can think of maybe an optical dye being the smallest, where you're really talking about the molecular scale, and then you can scale this all the way back counterclockwise to get to a microbubble, which is around 1 to 3 microns in scale. All of these can be used for imaging. They can be used for targeting, they can be used for therapy, or all three. And that's where and you can use these as sensors, okay? You can also break these into categories by materials. You can talk about organic nanoparticles, which might be composed of DNA, liposomes, those types of structures. You also have inorganic, which might be composed of metals like gold and silver, or they might be polymers. And then you've got carbon-based nanoparticles. And this is from a 2018 paper of how they categorize these. But these are kind of the playground you can use when developing a new nanoparticle or particle for the chemist and maybe the engineer, since now we're getting chemists and engineers, chemical engineering, as kind of the field that's the background expertise to do this. So when we start talking about intelligence, this is where we can then combine different functionality in the nanoparticles, including imaging, therapy, and targeting. But you have to then ask the question, what's the goal of designing the nanoparticle? And this is how I've been involved in this, starting especially at Michigan. I was really doing more of this at Michigan than here, maybe because the people I was collaborating with. But what I found was if you could imagine something that you wanted a nanoparticle to do, you could go to the top chemists in the world and they often could come up with a solution for what you were looking for. So first, you got to know what you're looking for. Then the chemist can, and you got to make sure that it has some relevance to some disease. Maybe it's cancer that you're trying to treat, and then you know what the targets are for how you're going to try to treat the cancer, et cetera. And then you go to the chemist and have them build it. So you can see that this requires some sort of collaboration. So you're asking the right questions, and the chemist can build it, and then you can do the testing and evaluation. So this is kind of the concept. A little bit complicated compared to just having a drug, but that's the concept. And I'll go into a little more detail and give you some examples of each of these as we go through. So this is a nanoparticle. I actually work with, I wasn't involved in this specific study, but I did use these nanoparticles that were called pebbles. And chemists are really good with coming up with acronyms, so I'll let you read the acronym. It's a lot easier to say pebbles. But this was a polymer, and in this case, they were designed to be 20 nanometers in size. And you could load things on the inside for imaging. In this case, you could put a dye or an MRI contrast agent, iron oxide, inside them. And then they would have an active agent that could be used for cancer therapy. So if you, in this case, photodynamic therapy. So you could either, you could activate a drug inside them, and then they would be active when they were triggered with a pulse of light or heat, something. And then you could put on the outside some sort of targeting. And in this case, it was an F5 target because they were targeting gliomas. And in the study, which I don't show you here, they did this in rats with gliomas, and they showed that this combined strategy, not just a nanoparticle, but there was a protocol, they were able to prolong the life of these rats compared to the standard protocol of just photodynamic therapy alone. Okay, so that's the concept. Now, taking this concept and then applying it to humans is a whole other story. Now, when it comes to imaging, you have to ask, what you would like to see, because you could look for dynamic signals like flow or clearance or accumulation over time, or maybe you're interested in a functional signal like the blood oxygen or oxygenation in tissue, or the pH or the glucose or protease, right? There's all kinds of things you could think of that you might want to see using nanoparticles as a contrast agent and as a sensor. And then it could also be, if you're targeting them, then you can get down to molecular imaging where you're seeing individual receptors or hormones or genetic signaling inside the cell, et cetera. So all those things are conceivable, they're possible, and in some cases they've been done. Maybe they've been done in vitro, maybe they've done in an animal. much fewer examples, I can say, in a human. So let's take another example. Another form of the pebble, this is called an e-pebble from about 20 years ago. And the goal here was to load the pebble with a special kind of optical dye. And this dye was sensitive to electric fields. So it's encapsulated dye. And then they program these nanoparticles to go inside the cell. And the goal was to image the electrical fields inside the cell. Now, that's very difficult normally. In fact, it had really never been done before this study, because you try to stick an electrode inside a cell, the tip of the electrode is bigger than the cell itself, so you can't really get a reference. You can do patch clamp. But what these were able to see were what they describe as lightning bolts coming from the mitochondria. So these would latch on to the mitochondria. And there were models before that, and now people believe that these electric fields are ways that the mitochondria and organelles are communicating. And these are huge electric fields, larger than the electric field across your cell membrane, was what they were describing. Okay, so these are all clever ways of using nanotechnology to see things that you might not normally see. I'm just giving you examples so you can start thinking about this and how it might relate to what you're working on. Okay. Now we get to therapy. So now we can sense things, we can see things, we can target things, but we also got a playground when it comes to therapy. We have to decide, where's the place of action? What type of tissue? Are you going to be intravascular, intracellular? These are all questions you can ask, and you can design your nanoparticles to obey whatever you design. Then the question is, what's the method of therapy? Is it some sort of chemical reaction? Is it going to be heat? Is it going to be radiation? In some cases, deliver radiation to the plate. Is it going to be genetic? And then the mode of action could be, the goal could be to just simply destroy, like a tumor, kill the tumor. But sometimes all you got to do is suppress it and just leave it there, and the person can survive. Or you can modify the code and maybe even turn it back into a normal cell if we're talking about cancer. And then the delivery, right? So you could have the delivery of the drug be passive. It could be instigated by a chemical coming in contact with a certain chemical, or it could be promoted by a pulse of energy. It could be an ultrasound pulse that releases the drug. It could be a pulse of light at a certain wavelength. It could be a pulse of microwaves. And so these aren't just ideas. All of these have been demonstrated in vitro, in an animal, et cetera. So all these things are possible down at the nanoscale. Here's an example of beads that release drugs at a certain temperature. So at body temperature, they're encapsulated, they're stable. You increase their temperature to 40 degrees and they release their contents. So there's lots of ways you could heat the tissue and do this with sound, light, electricity, et cetera. There's lots of ways you could do that, or just heat normally. Here's another example. These are called, it's not necessarily heat that's activating these carbon nanotubes, but it's actually a single pulse of light makes them pop, and it causes destruction. And in this paper, they show that the cells near this destructive event, when they pop, basically those cells die in that neighborhood. Okay, so I'll give you two examples there of how that might work. Then you get to the targeting. All right, and targeting is pretty much done on the surface. And this is an article from back in 2003 of all the different things you can put on your nano platform for targeting specific receptors, cells, to go inside cells. And there are often lookup tables that you can use that are used routinely in drugs and fluorescent agents, et cetera. And the same sort of principles can apply to the nanoparticles. And I give you just a few examples. This is from things that we do with photoacoustic imaging. That's why in this case, these nanoparticles on the left all absorb near-infrared light because they're designed for photoacoustic imaging. But you can also see the targets they were used. And these are specific to cancer targets. You could look at injury, you could use ICAM, and there are many different things, again, you could look up depending on your condition. Okay, so now let's give another real world example of how we could use this. This is from Georgia Tech University that developed these gold nanoparticles with glycol chitosan coating on them because they wanted these particles to be picked up and trafficked through the lymphatic system. That was the goal. Because they wanted to have a sensor, a contrast agent that could detect specifically metastatic cancer. And to be able to do that, if in metastatic cancer, the lymph nodes, sentinel lymph nodes are compromised compared to non-metastatic breast cancer. So if they could come up with a nanoparticle that could specifically see that, that could be kind of game-changing in terms of contrast agent. And I won't go through the optical properties of this agent, but I will just say that they use photoacoustic imaging to do this, and you can kind of see the results there. So in the naive control, so they wait 24 hours after these nanoparticles are picked up by the lymphatics. They get deposited into the lymphatic system, into the sentinel lymph node. And they have a very specific signature when they're encapsulated by the cells that take them up. And they're optically change their properties. So it's very specific, the signals that are coming from, in this case, the sentinel node in a mouse. But you can see that in the metastatic case, there's far fewer nanoparticles getting into the node. So you can see statistically that they could identify that that was metastatic breast cancer. Okay, so there's an example. When we get to the brain, there's a whole other barrier we have to consider, and that's called the blood-brain barrier. So people have wondered whether nanoparticles can facilitate drug delivery to the brain, which is kind of an oxymoron because more than 80% of drugs that would normally get to other types of tissue cannot even get into the brain, and that's why it's so difficult to come up with new drugs for the brain. But in this case, people were coming up with approaches that are listed on the right to deliver things into the brain. And this blood-brain barrier is kind of a tight junction of cells that keep things out of the brain. It's a protective barrier to keep things from your blood getting into the brain, et cetera. And that's what makes it so difficult. But it also means that your brain needs to be protected from these foreign particles as part of its protection mechanism. So let's just say let's try the magnetic nanoparticle approach. Can we use a magnetic nanoparticle to improve delivery of a drug into the brain? And in this case, we go back to rat gliomas. Now, you'll notice a theme here that I'm often using gliomas as kind of the model, because the few cases where you've actually seen these exotic nanoparticles used in humans are for fatal diseases like gliomas, where there's not any other option. And so people are desperate and they're ready to try anything. So these types of things have been used in those cases. But in this case, what we're doing with the magnetic nanoparticles is simply allowing them to circulate and then using a magnet placed over the area where the tumor is and to increase the dose or load of that drug to the tumor. That's the idea. Now, this concept has been used in humans. I'm not sure necessarily nanoparticles, but for drug delivery by having magnetic properties and using magnets to increase the dose. That is something that is used and has been used to improve delivery of drugs, this concept. So that's kind of an example. All right, so now the question is, do we see this being used in humans? And if so, where and why not? So these concepts, a lot of the studies I just talked about were from 20 years ago. And despite the promise and a lot of investment, like NIH had got billions of dollars around 2005, built a program, nanotechnology program, and I'll come back to that in a second. But for any drug, including nanoparticles, this is the development process that's normally required for FDA approval. So you can kind of see that's 18 years. And if it's for imaging, you're not usually going to take much risk if you're just using something as an imaging modality. So company isn't going to be too enthusiastic to develop a new contrast agent, go through any part of this process with the idea of coming up with an agent that might be used for, for clinical imaging or something. It's usually for drugs. And there's not a big incentive for companies these days to kind of go through this process, but I am putting that up there to get an idea of what's required. But another kind of turnoff for companies might be the fact that nanoparticles in general are toxic. NIH did several studies in the 1990s trying to treat mental disorders or neurologic disorders with nanoparticles, and there were a lot of side effects during those studies, and they had to abort a lot of the studies. And since then, there's so many articles and papers that talk about nanoparticle toxicity. This is one from 2008, but you can find so many of these, but you can kind of get an idea of the kind of things that the nanoparticles can do just by looking at that list, and I'll come back to that in a second. Oh, that's the paper, drug delivery and nanoparticles, applications and hazards. One slide specifically on graphene nanoparticles, carbon-based nanoparticles, promise and perils. There's quite a bit of research because graphene has a lot of interesting electrical, magnetic properties, mechanical properties, carbon-based, and giving you examples there of using, there's electrodes you can insert in the brain for recording, doing electrophysiology, and interfacing with neurons and stimulating neurons. So both recording and stimulating. There's lots of papers demonstrating this. A little bit different than optogenetics, which uses light to activate specific types of neurons. People have been trying to work with graphene and carbon-based nanoparticles to kind of get a similar effect and use some sort of external energy source to interface with the brain. The problem is, again, the toxicity of graphene is well known, well documented. So other than, doing things in an animal or in vitro or brain-computer interface, which is a lot of where this technology is, it's hard to see how this will evolve into an effective safe treatment, but that would be part of an open conversation to figure out how to do that. And then finally, here's more specifically talking about the neurotoxic effects. The other was more general toxicity. Many articles talking about these neurotoxic effects, and the next one is more of a graphic representation. So we can look at that. These are mechanisms and factors. You can see the kind of things that nanoparticles can do that may not be good. Tumor promoting, immune destruction, deregulation of cellular energies, promotes inflammation, apoptosis, necrosis, genome instability. So these are all reference papers. These are related to reviews, and you can kind of see the kind of things that the properties of the nanoparticles that are important here: the size, the geometry, the material, the electrical properties, and the charge. All of these will impact how reactive it is to the tissue. Okay, now I want to just pause there a second. So about 10 years ago, the director for the Center of Nanotechnology at NIH was here at the university, and I kind of asked him, like, what's been the biggest accomplishment of the nanotechnology at NIH, and like he kind of like paused and took a while to answer, and because I kind of pointed out that these things are generally toxic and we don't really see many success stories, and he basically agreed, and he said that the main accomplishment that he could attribute was reducing side effects of chemotherapy. So liposomes, the encapsulation of the drugs that are often used for chemotherapy have dramatically reduce the side effects of those. And so that's encapsulating a drug inside the liposome, which is that. So that was kind of his response. So just something to think about. Okay, so now that we've kind of established what's possible and some of the perils of nanotechnology, now we're going to kind of hand things over a little bit more towards the engineering side of things to see what's possible and expand the mind of what you might do with a nano robot or nano society or whatever word you want to use. So this is Charles Lieber's work at Harvard University from 1999, where he was able to build actuators on the size of 100 nanometers. You could actually move things around with these nanoparticles. And these are little tweezers that could then be controlled. And that's back from 1999 in Science. And why is that important? Well, a few years later, he got a lot of funding. You can kind of see who's getting funded from DARPA, Navy, Air Force, a little bit from NIH, et cetera, and a lot of companies. And you can kind of see what he's talking about here in terms of the ability to make nanosensors and interfacing with cells in the body on a large scale, and essentially building transistors or even computers on a nanoparticle. Okay, so this is a combination of electrical engineering now, chemical engineering, et cetera. This is kind of now 25 years ago. So if you think about that, term liquid computing. So you go forward 10 years, and they came up with a way to, and there's a lot of backstory to this and getting the geometry and the structure right, but they were able to develop things that lodged and held on to cells and could stay there for long periods of time. You kind of see that there. And these ultimately were transistors that could send and receive information. So lipid-coated silicone nanowires, and that geometry became important because they had a hard time of getting it into the membrane and sticking there. But that's what it kind of looks like, and that's from 2011. And many patents are related to this. And now this kind of spawned a whole field. Of course, he continued in that field. And if a lot of people don't know, Charles Lieber was arrested in February 2020 for some things he was doing with China. And I'll just put it at that. You can kind of look into that. But he was, at that point, I think, head or director of biochemistry at Harvard when he was arrested. But I think he was collaborating maybe a little too closely with China. I'll just say that. Okay. Anyway, but he definitely was a pioneer in this field. And shortly after that, we started to hear words. Of course, 5G comes a little bit later, but the Internet of Things, Internet of Bodies, all this lingo and terminology that mainly comes from United Nations, World Economic Forum, Davos, that kind of group. But these are buzzwords for industry. And so it's really important to recognize that the kind of technology we're talking about, the emerging technology, the nanotechnology, and the surrounding required infrastructure, these are multi-trillion-dollar industries. And if the goal is to make people healthier, we need to establish that. But a lot of the things in the literature are engineering-related journals. And so what you're seeing here is an article, I think, from 2015, and this is another term, Internet of Bio-Nano Things, of being able to build computers on nanoparticles, communicate, two-way communication with that. That's the concept, and these things have, to a large part, been demonstrated to be possible. And this is a review paper specifically on the Internet of Bio-Nano Things, of being able to use, in this case, a bacteria, an engineered bacteria-based biological transceiver. Now, this has some interesting ideas. What if you could just send these into the body and they could communicate as sensors back outside the body for sources of information? You can imagine that they could communicate all kinds of information that could be very valuable to know what's going on inside your body that you wouldn't be able to do. Now, there's a lot of caveats to that, because how do you do that safely? What's the impact, et cetera? But the concept is interesting from the standpoint of sensing and communicating with the body. But let's continue. So here's now an example of the same sort of idea, a nano machine. That's what the architecture looks like, with the idea now of communicating from outside the body. So you've got your phone, your laptop, and then that can go to the internet. So now you can get a lot of information transfer about things that could be on your body. I'm not talking too much today about wearables, but all these things work in synergy. These are different types of emerging technologies, but coming from the outside, coming from the inside out. And so you have this network architecture now, intra-body application. And those are the types of communications that are possible, that can communicate with a wearable, that can then get to the internet and then go anywhere you like. To a little bit closer look at the types of molecular communication, calcium communication, molecular motors, which I find to be a really interesting field, how all these little structures biologically develop ways to transport themselves, and then bacteria, so chemotaxis, and then hormone communication. So those are 4 popular things. And there's a quote from that article promoting it. And then from that same article, the idea of building artificial biological cells for networking. So these are entire cells that could function artificially, I don't know, biological nanostructures or whatever. I think you guys get the idea of what the concept is. Now let's take it and look at the infrastructure. So this is called the wireless body area network, well known and understood in the engineering field. And the communication protocols for the devices that I've been talking about, from nano to wearable to outwards, was established in 1995 by IEEE. And those are the frequencies, the protocols, et cetera. So that's 30 years ago was when the protocols were developed for the communications of these devices. And even when I was in graduate school, we had wearable ECGs. They weren't really promoted like they are today, but we had wearable devices back then. So it's really not so much new technology, other than it's just more in your face to the public technology, and being promoted by industry. A little bit more on that communication, on how the W-band and interfaces with the nanoparticles and nanophones, and I don't even know what all these terms are, but maybe some sort of promotion of the technology, but you can see that. And then the bands and the frequencies that are involved with communication. And then it's much more elaborate, because the internet, the nano things here in the bottom right, bottom, is just spokes from a much larger configuration of AI and communications and other things coming together. So somebody sat down and put a lot of time and effort into thinking about this kind of a world, whatever kind of world you want to call it. Okay, so I'm kind of winding down here, but and I said I'd go to 7, so I got 5 minutes. I was going to play from a video, but I don't have to. This is an example of manipulation of DNA, and the chemical engineers have great expertise of manipulating and building things. And here's an example from DNA from Caltech 2006. And this is part from also from a TED Talk, which I'll link to. Maybe I'll play a couple minutes to 7 just so you can hear. But it really goes through all the things that we just talked about, especially the last several slides being developed at the university. And it's a TED Talk from 2013, so keep that in mind. But these are how nanobots, that I've been talking about, how they're promoted as potential benefits. And you can kind of just read all the different possible things there. And then these are kind of the challenges, some of which I've already kind of like mentioned here. So I think I'm almost to the end of this. Maybe before that, I'm just going to add one more thing, because I know Anna was asking about transhumanism. So I want to just put this up here because it's very much involved. A lot of the things I just talked about, it does connect with transhumanism, which is a religion. It's a religion that United Nations, World Economic Forum, they promote. So the idea of merging man with machine and leading to immortality. So many of the Davos men talk about this concept of merging man with machine to become immortal. These are the things when you read their books and listen to their presentations, they talk about. So there is a connection with all these things, engineering and connecting to biology and kind of the merging with machine. So having said that, so this talk, let's see here. So this is the University of Bar-Ilan in Israel. And this is Professor Ito from 2013 talking about, let me just see if I'm connected to, I don't know if I am, I'll try it.
Of them in a tube in a single reaction. Now, since 2006, several researchers, very talented ones, have been expanding
Sorry.
And these researchers also gave us computer-aided design tools that enable everyone, very, very simply, to design objects from DNA.
Oh, wow.
So these CAD tools, amazingly, enable us to focus on the shape we want, forgetting the fact that these structures are, in fact, assemblies of molecules. So this is, for example,